Hello, and thank you for joining me. Today I'll be talking about the research that I've been doing on the convergent evolution of limblessness in squamates. But first I'll tell you a little bit about myself. I grew up in Chicago, which is a city in the US that's about 3 million people, and if you've heard anything about it, you've probably heard that it's cold. It's not a very hospitable environment for herpetofauna. I didn't actually see my first wild squamate until I was 10 years old when I went on vacation to Florida. Though there aren't a lot of reptiles in the city, there's still actually a lot of green space, and I got a good amount of nature. I also had access to some amazing museums. I went to the Field Museum and looked at dinosaur bones, and also to the Shedd Aquarium where I saw fish and dolphins. I grew up being fascinated by nature and went on to study biology in college at the College of Wooster. I did my senior thesis project on speciation in island-dwelling snakes, and though I found that systematics wasn't for me, I became hooked on research. I interned at the Field Museum in Chicago, and then went on to get a master's degree from Virginia Tech in paleontology. As a kid, I asked a lot of big questions, and that hasn't changed. A few are, how predictable is evolution? What might life be like on other planets? What was life like in the deep past? And are humans really that special? We can get insights into all of these questions by studying convergent evolution. This is when lineages that are unrelated and unalike evolve to be similar. One example is the shark and the dolphin. Their ancestors look very different. One looks like a fish and one looks like a rat. But they both converged on this morphology, this torpedo-like body shape with pectoral fins, a dorsal fin, and a forked tail. We can use convergent evolution to infer things about life in the past. Sharks and dolphins both eat agile prey and swim very quickly. So when we look at this extinct marine reptile called an ichthyosaur, we can infer that they also ate agile prey and swam fast. Studying convergent evolution, or the absence of convergent in certain lineages, can reveal a lot about evolutionary constraint. One example is flippers. They've evolved several times in mammals. Moving around in a watery environment limits the forelimb shapes that will be successful. A paddle-like flat limb will do much better at moving you around in water than long, skinny fingers. This is what we call an ecological constraint, so the environment constrains the options for how things can evolve. One interesting thing to note is that marsupials have not evolved flippers. One hypothesized explanation for this is that there's a developmental constraint that prevents marsupials from evolving varied forelimbs. Newborn marsupials usually crawl with their well-developed forelimbs to the nipple inside the pouch. If a newborn marsupial was born with a limb that couldn't grasp fur and couldn't climb, it wouldn't be able to survive long after birth, even if the limb was perfect for swimming. As you can see from this picture here, the forelimbs of this baby wallaby are well developed compared to the small hind limbs. Studying the similarities and differences between clades that have or have not evolved convergently can tell us a lot about how evolution works and how it's constrained. To find out how predictable evolution is and what constraints are most influential, I decided to focus on a frequently evolved body shape. We know it well from snakes, but actually a lot of different lizards have evolved the snake-like body form seen here. All of these animals may look like snakes, but if you look closely, you can see all of them have remnants of tiny limbs. This body type has evolved many times across the squamate tree. In fact, at least 26 lineages in Squamata, all across the tree, have evolved this limbless body plan. 7 different major clades have evolved it. But one clade has evolved it more often than any other. Skinks have evolved limblessness at least 19 times. Why go limbless? Limblessness is hypothesized to be an adaptation for burrowing. Going underground is a great way to hide from predators, find burrowing prey, and hide from harsh weather above ground. And if you're traveling in a burrow through sand or through leaf litter, and you have the ability to propel yourself forward by undulating your body, limbs might actually get in the way. Many legless reptiles have converged on traits that are advantageous in a life underground. In addition to limb reduction and loss, many of them elongate their bodies and develop traits on their head, such as an overbite, reduction in eye size, fixed eyelid, so they can't blink like snakes, reduced or absent ear hole to reduce the amount of sediment that gets into their body. They often reduce their tails if they're a burrowing morph, but for those that remain overground and maneuver through leaf litter, they actually seem to lengthen their tails more often than not. And then many of them often lengthen or reduce the size of their internal organs, such as their lungs. I use the term limbless a lot, but it's actually pretty hard to define. Many limbless species, even some snakes, retain rudimentary limbs, so they aren't completely limbless. And some, like Lerista edwardsi here, have almost non-existent external limbs, but still technically have internal limbs. If you look at them, you may not think that they have limbs, but if you look really closely, you see this projection here that looks like a claw. And if you look at their CT scan, you can see the humerus is definitely there. Another example is this one here. We see on the left the skeleton, and then on the right the pelvic girdle. And you can see these two blue nubs here. Those are actually the remnants of the femur. So they technically have limbs, even though they are nominally limbless. In fact, the vast majority of squamates still retain a girdle element, whether a pectoral or pelvic girdle. This means that most squamates aren't technically all the way limbless, as they still retain some signs of having limbs. One group that is particularly well suited for studies on limb loss is the Australian genus Lerista. It's a skink genus that is comprised of about 100 species, and they range from penadactyl to completely limbless. But actually, most species in this genus have transitional morphologies somewhere in between. This is a phylogenetic tree of Lerista illustrating the diversity of limbs found in this genus. I've used symbols to represent the category of limblessness they fall into based on digit count, or limb proportions. For example, if a species has 2 stars next to its name, it is pentadactyl. If it has a diamond followed by a circle with a black dot in it, that means it has 2 digits on each hind limb and no external forelimbs. I'll be using this scheme throughout the entire presentation, so make note. One remarkable thing about Lerista is how frequently limbs have been completely lost. There are at least 3 different instances of complete forelimb loss in this one genus. And this has happened remarkably quickly. It's estimated to have happened within about 4 million years. I'd like to take a moment here to acknowledge the traditional stewards of the lands upon which my specimens were collected. Now, I'm not the first scientist to recognize how special Lerista is. Other researchers have found relationships between elongation and limb reduction, and a number of other characteristics. Most previous studies have used presence or absence data, phalangeal counts, and 2-dimensional body proportions to study external morphology. How do internal structures change during the transition to limblessness? Are internal structures subject to relaxed selection? In other words, if a limb support structure no longer has a limb to support, is there going to be selection to maintain its structure? Will the structures disappear and deform rapidly with no maintaining selection? Will they stick around? Do internal structures change in a predictable way, or do they change at random? I decided I would learn most about evolutionary constraints by studying the pectoral girdle. It's a 3-dimensional structure comprised of a complex of bones that support the limb and serve as muscle attachment points for neck and shoulder muscles. They may reveal differences that are more difficult to capture using simple metrics, and they may be under different selective pressures depending on whether or not there is a limb. Here's a figure depicting the lizard pectoral girdle. Their pectoral girdles are a bit more complex than ours are. They have clavicles and sterna and shoulder blades just like we do, but their shoulder blades are fused to a bone called the coracoid, so they have what's called scapulocoracoids, and attached to those are cartilage structures called the epicoracoid and the suprascapula. They also have this bone in the middle called the inner clavicle. To look at the pectoral girdles of Lerista, I used computed tomography or CT scans. It's basically a three-dimensional X-ray. I want to take a minute to comment on how cool they are. So here we have an X-ray of Lerista stylus done in 2011. This was an investigation of their pectoral girdles, and we can see that their result are these two stick-like structures. Not very distinct, we can't tell what they are. Looking at a similar specimen done with a CT scan, we can see that their pectoral girdle is pretty complete. All bones are represented. CT scans can reveal a much more complete picture of the pectoral girdle, and they come with other advantages as well. We can use 3D objects for digital analyses, which is what I'm using for my project. I'm using geometric morphometrics. This is a way to simplify complex shapes using homologous landmarks. You place landmarks on each specimen on features that they all share, and then you standardize the size and the orientation and use a computer program to compare the shapes. The results end up looking like this. A PCA analysis, or a principal component analysis, is basically an analysis of similarity. For our data look like this, we have a circle group and a star group. We can see the circle group is clustered very closely together. This means that they are more similar to each other than the spread out star group, which has a much wider area of morphospace, and that means that they are less similar to each other than the circle group is to each other. Here's what I predict my results will look like. I'm hypothesizing there will be relaxed selection, so species that don't have any forelimbs won't have any selective pressures to maintain the structure of the pectoral girdle, but the species with pentadactyl and tetradactyl limbs will have selective pressure, keeping the integrity of the pectoral girdle. So those will cluster together and be more similar to each other because there's a functional reason for them to stay looking the way they do. But for those without external limbs, there's no reason for the pectoral girdle to stay the way it is, so I predict that the pectoral girdles of those without limbs will be more spread out and less similar to each other. And here's what I found. Each symbol here represents a specimen, and we can tell what their forelimb morphology is based on the symbol. Remember, pentadactyl is represented by a star, no limb is represented by a circle with a black circle in the middle. I've also used colors for this to represent where each specimen fits in the morphocline. So I've ranked them all based on not only their forelimb morphology, but their hindlimb morphology and limb dimensions as well. Basically, this gives us an idea of where they are in the transition to limblessness. The closer a species is to red on the rainbow, the more divergent it is from the original five-fingered ancestor. From this, it's clear that there is a relationship between how limby a species is and their pectoral girdle morphology. We can see this especially in the PC1 axis, where we have on the right the tetradactyl and pentadactyl species clustering over there, and on the left we have the ones with no limbs, and everything else sort of in between. We see it's in rainbow order, so we know that it's in order of the morphocline. However, we don't see the spread in the limbless section that I expected. This means that there's probably some sort of selective pressure that's keeping these pectoral girdles in the same sort of neighborhood in terms of morphology. One thing that we can tell is that in general, pectoral girdles have changed from being tall and narrow and very curved to sort of more short and flat. I found some other interesting patterns that have to do with how the pectoral girdle and their associated features are lost. Here we have Lerista allinae and Lerista edwardsae. Both have these small little blue bones that are all that's left of their arms. That's their humerus. But they also have these little holes here. These are the coracoid foramina, and these are holes through which the nerves of the arms pass. Now we can see Lerista stylus and Lerista praepedita on the right lack both of these structures. And I couldn't find a single specimen that only had one. They either had both or none. This indicates that there's some sort of process or some sort of constraint that keeps these two changing together. However, I noticed a disconnect between the degree of limb reduction and the degree of girdle reduction in some species. We have Lerista apoda on the left and Lerista amylase on the right. Both of these species have no external limbs whatsoever. We can see Lerista apoda's pectoral girdle is kind of just indistinct mush, whereas Lerista amylase, we can see all of the bones are accounted for. It has a complete pectoral girdle. Lerista apoda, in fact, this is just my best guess for the identity of the bones. It's really, it's anyone's guess as to what these are. And it's very interesting that the same degree of limb reduction would lead to such a different degree of girdle reduction. This means that the two things are not entirely connected. I noticed an interesting pattern in regards to the influence of phylogeny on pectoral girdle morphology. Here we have two closely related clades that represent two independent losses of forelimbs. On top we have Lerista carlschmidti, Lerista carpentariae, and Lerista stylus. And then on the bottom we have Lerista cinerea and Lerista amylase. The top clade tend to have straight or bowed upwards clavicles, and their inner clavicles tend to be rather short, and the sterna tend to be fairly indistinct. The bottom clade have very distinct bones. They have arched clavicles and inner clavicles that overlap with the sterna. They also have distinct scapulocoracoid cartilage elements, whereas the top clade does not. This suggests that phylogeny and phylogenetic history plays a role in constraining how pectoral girdles change throughout this transition. So far, we can conclude that CT scans should be used instead of X-rays for future work on pectoral girdles. We can conclude that overall morphology of the pectoral girdle has evolved in a very similar way across Lerista lineages. There doesn't appear to be much relaxation in selection. The girdles are changing in a similar way, even in highly derived limbless species. Specific aspects of pectoral girdle reduction may be clade-specific, so there are some differences between the clades. And pectoral girdle reduction appears to be happening in associated with limb reduction, but it's not a perfect relationship. One piece of data that proved to be frustrating to get was the position of the pectoral girdle. One basic question about the evolution of limblessness is whether or not the neck gets longer during the elongation during this transition. And the position of the pectoral, because it is right in between and serves as the division point between the neck region and the trunk region, by locating the pectoral girdle, we can locate the neck-trunk boundary and therefore figure out how long the neck is. But what if we look at species that are more elongate? This is Lerista stylus and Lerista apoda. And if we look at hypopophyses in blue and the pectoral girdle in green, We see that the pectoral girdles are at vertebra number 8 and vertebra number 9, which means there would be slight neck elongation. But if we look at the hypopophyses here in Lerista stylus, we see they go down to vertebra number 7, which would sort of fit with the model we had with the other specimen. But with Lerista apoda, the hypopophyses actually extend beyond the pectoral girdle. So those features may be inconclusive. What about overall vertebral morphology? Most lizards have body regions with distinct vertebrae, just like humans. If you pick up a vertebra, you can tell which part of the vertebral column it came from, based on its morphology. Vertebrae are complex shapes. Can you think of a method one might use to quantify, simplify, and group such complex shapes? Geometric morphometrics, of course. Geometric morphometrics can be used to find boundaries between these regions, and have been in the past, even with snakes. But do these boundaries, based on geometric morphometrics, match up with other neck trunk boundary markers in legless lizards? The short answer is no. I performed a geometric morphometric analysis on the vertebrae of Agamodon and Guliceps. Here we see in green the pectoral girdle sits around vertebra number 3, whereas the hypopophyses in blue go all the way down to vertebra number 14. Now the black circles on this graph here represent the PCA scores, or the principal component scores. This is based on their similarity to each other. And we do see that vertebra number 3 is most similar to vertebra number 4, and vertebra number 4 sits in between vertebra number 5 and vertebra number 3. So we see that there is sort of a gradient in similarity between the vertebrae, but we also see on the PC2 axis that there's sort of a shift in morphology that happens. This is where the neck trunk boundary is according to the geometric morphometric analysis. I've put it here, around vertebra number 7 or vertebra number 8. And this is not where the neck trunk boundary is according to the location of the last hypopophysis or the location of the pectoral girdle. I also performed one for these other species, Lamprophilus guichinoti, a skink that is pentadactyl, Lerista stylus, which is a skink without forelimbs, Lerista apoda, a completely limbless skink, and then yes, Agamodon and Guliceps. And for the pentadactyl skink, we see a match between all 3 of these neck trunk boundary markers, but for Lerista stylus and Lerista apoda, they don't match up. Lerista stylus, there is a matchup between the hypopophyses and the pectoral girdle, but not the geometric morphometric neck trunk boundary. This may mean that whatever is patterning the neck trunk boundary based on geometric morphometric scores or overall vertebral morphology is changing, or is, I guess, staying the same, and the other 2 are changing in tandem for a bit, but further along in the transition, they too separate. This is a very small sample size, so it's nothing to get too excited about so far, but I think there's some pretty interesting implications here for developmental biology. All 3 of these neck trunk boundary markers must be patterned by something different in development. And the fact that they're disassociated in the evolution of limblessness is very interesting, and means that they may be patterned by different mechanisms, and that there may be more than one neck trunk boundary. In conclusion, the neck trunk boundary is still difficult to define in limbless taxa. It is still unclear which marker best represents the boundary, and therefore it is still difficult to tell whether the neck was elongated during the transition to limblessness. However, this may not be the right question. These results suggest that each neck trunk boundary marker may have a unique patterning mechanism, which means it may be more important to resolve how these neck trunk boundaries are patterned before we approach the neck elongation question. So what's next? I intend to continue exploring the neck trunk boundary in legless lizards, and I'll be presenting more results at SICB on the neck trunk boundary stuff, so if you're going to that, check it out. In the future, I want to incorporate development and genetics into my work. I really want to find out how legless lizards in different groups develop, so not just skinks and not just Lerista. I want to find out what genes are involved in these morphological changes, and whether there are genomic factors that predispose some groups to limblessness. I have some other projects going on that I'm really excited about. One of the scans that I was working with was of a nearly legless lizard that had a skeleton of a nearly legless lizard inside of it. Based on the ranges of species, limbless species in the area, it looks like this might be cannibalism. And that would be really exciting because this species is critically endangered and we know absolutely nothing about its diet. So one of my projects for the next few months is to figure out what the species inside is so we can figure out whether this is predation or cannibalism. I've also become interested in the switch between viviparity and oviparity. There are two species of Lerista that give birth to live young, and I've CT scanned the babies pictured here, so I'll be able to tell a little bit about their development and how baby Lerista grow. Unrelated to these, I've also been involved in increasing awareness of disability rights in academia. I have ADHD, and I founded a support group for neurodiverse biologists so we can inform people in our department and across academia about disability rights, especially for graduate students. And with that, I'd like to thank a whole bunch of people. First, I'd like to thank people involved in collections. The collections managers were absolutely essential for my project. They helped me get my data, and I wouldn't be able to do it without them. I'd also like to thank my master's advisor, Michelle Stalker. I'd also like to thank Ed Stanley for giving me access to what I needed to collect my data. I'd like to thank my disability learning specialist, Amanda Jackson, and I'd like to give a special shout out to people involved in MorphoSource and other open access resources. They allowed me to do my project. I'd also like to thank my family and my partner, Patrick. And finally, I'd like to thank the Australian Herpetological Society for hosting me with this talk. If you'd like to contact me, if you'd like to collaborate on anything or have any cool ideas, please let me know. Thank you guys so much for listening.